I just wanted to acknowledge before I give this talk that I'm pretending like it's my work, but it's really not my work. These are the main group of people that I've been working with. This is Yi Zhao, who's at IUPUI, Bingkai Wang, who's now at UPenn, Bo Hao, who's a PhD student at Johns Hopkins, and Rossi, who's at UT Houston. So we have a really wonderful group that meets Friday afternoons, and we talk about these sorts of things. And what we tend to think about is, one of the things we tend to think about is how to generalize regression, not just in the field of neuroimaging, but generically outside with methods that could be used in neuroimaging, but then could be more broadly applied outside of neuroimaging. And you know, it's worthwhile sort of thinking about the classic treatment of regression, or at least how I like to think about the classic treatment of regression, where you think about a linear subspace that spans your collection of regressors, and then you project your observed data onto that linear subspace. That's the classic variation of least squares. And I couldn't find a picture of him, but the original variation of least squares was invented by Legendre, a French mathematician who invented it in the late 1700s, I think, and then or early 1800s. And then Gauss, of course, I think famously said, "Yeah, I did it. I just never published it," as well. But that's not the only way to think about regression. There's several different ways to think about regression. There's, projection is the classic way that gets taught in most statistics classes, or you could think about a likelihood type approach where you assume some normality assumptions and a correct mean model and maybe independent errors or something like that. I like to actually teach that you can iteratively apply regression to the origin and come up with multivariable regression, or iteratively apply any collection of regressors. And it's interesting to note, this is an interesting way to think about it as it relates to pre-processing, because you can think of pre-processing as iterating through a series of projections for at least the linear pre-processing steps. And Martin has a really great paper about why you should maybe think twice about that. It's a conditional estimate based on the joint normality of the two, the regressor and the response, assuming joint normality. And most statistics classes, I think, teach that regression has some dependence on Gaussianity for inference, but that's not true at all. It only requires a first and maybe a couple assumptions about the second moments and maybe some sampling-based assumptions, and then you recover not only regression, but basically all the typical inference that we teach under very mild conditions. And there's all sorts of generalizations. And I want to point out one in particular that was that one application in neuroimaging that came out quite some time ago that's quite relevant to what I'm going to talk about today. This PET and fMRI paper from Worsley, Colleen, Friston, and Evans. What they were really thinking about was transforming the response, transforming an image-based multivariate response so that the linear model fit well, not unlike partial least squares does. And there's many ways to think about it, but I think I just wanted to mention that what I'm saying is just a reinvention of some stuff that's already pretty well trodden territory in this field. So I'm going to assume that I have data that come in the form of a connectivity matrix. I think in this crowd, I don't have to belabor how one comes up with that, but just to cover it briefly, let's assume we have some fMRI data. We turn it into a big green block by stringing out voxels along the columns and time along the rows, and then we grab columns somehow, either by taking a regional parcellation or by taking seeds or something like that, and then we take the outer product of the collection of black squares together. We take the outer product to form a correlation matrix and get something like this. That's necessarily, from a matrix standpoint, positive, definite, and symmetric. And what I'd like to think about is instances where this quantity is thought of as the outcome. And we don't actually have a ton of statistical techniques for positive, definite, symmetric matrix outcomes, where we measure one such positive, symmetric, definite matrix for every unit available in the regression. So there's there's a literature, there's kind of a blossoming literature in the statistics about kind of non-standard outcomes. And we decided to narrow down to two approaches. One was just to ignore the regression problem first and think of ways to decompose the connectivity matrices. And we thought of doing that with some, a variation on PCA called joint PCA. And then the second is much more related to that paper by Worsley and Friston and Pauline and Evans that then tries to find the best regression model that fits to the, find the best rotation that fits the best regression model. So I think in neuroimaging, people are quite familiar with common PCA because of the sort of dominance that common ICA or group ICA has in this field. But let me just talk a little bit about common PCA. So imagine if I was too lazy to draw data, fake data points, and wanted to talk about PCA of two regions. So imagine you were ignoring time, and you were just plotting the time points as if there were a bunch of IID data points between two regions, and then they kind of fell in an ellipse. The common PCA assumption that we'd be making in this work is that the sort of forgetting about noise, the main angles of the data are consistent across subjects, whereas the length of the angles may differ by subjects. And that's, of course, not the only assumptions that one necessarily need make. You could, of course, assume that the angles vary by subjects, but that the sort of engagement differs by subjects. That this assumption, I think, is more consistent with what we're thinking about in neuroimaging, just because it's sort of, you might think by analogy, it's that the networks are the same across people, but the network engagement is differing by person. Whereas this would sort of assume that the networks differ by people and the network engagement is consistent. And I think the first assumption is probably more relevant. And when we thought about common principal components of a connectivity matrix, we started with a simple insight. And of course, the end version of the result was much harder than this. But here's the simple insight that imagine if you assume that the average or population-based expected value of a connectivity matrix followed constant rotation. So the gamma, the exterior matrices are the same, whereas the eigenvalues are unique to a subject. And then if you were to be able to fit a model like this, you could then model something like the logarithm of the eigenvalues with a regression model. Well, if this model holds, then the average, the expected value of the average holds in this way. And then you could do some method of moments approach. And so that's what we took to calling semiparametric, or a generalization of this as semiparametric common principal component analysis. And this builds on some well-known work from a statistician named Bernard Fleury, who sadly died quite young. He was in his early 40s and tragically died. But he was a really wonderful multivariate statistics researcher. His approach primarily worked on likelihood-based approach using Wishart distributions. So instead, ours uses partially common eigenvalues, which Fleury also did, but based on moments-based approaches. So we assume that only some of the connectivity matrices are consistent across subjects, and the rest are idiosyncratic or unique. And so then we estimate the number of common components, because if you're going to have common parts and non-common parts, you need a way to decide what's common and what's not. And to do that, we use a measure called the deviation from diagonality, which is from Fleury. And then we established statistical asymptotic conditions as number of subjects goes to infinity, but also as number of time points goes to infinity for fixed number of subjects. The latter, of course, requiring really fairly strict assumptions that aren't that much weaker than distributional assumptions. But the result is the idea of using semiparametric common PCA, and then you fit a regression model to the idiosyncratic eigenvalues where everyone has the common networks. We are able to show that the approach does do the sorts of things that you would like it to do. For example, if you were to take the HTTP data and take task data inside the motor network and do this kind of common PCA and look at what some of the common components are, they would sort of loosely organize with respect to the task, and their sort of homuncular organization would agree with what we know about M1, for example. However, the problem with this approach was that it turns out that if you find good directions of variations in subjects disregarding the regressors, in many of the examples we looked at, they were fairly unrelated to the regressors themselves. And that's the hope that you have whenever you do a sort of separated principal component analysis. If you principal component decomposition your regressors, if you have a high dimensional regressor and then put it in a regression model, or the outcome and then put it in a regression model, even if you do a fancy technique like this, you're still hoping that the directions of variation that you find in the decomposition separated from the regressors is the interesting directions with respect to the regressors. And for most of our problems, it was turning out that was not true. So the approach, while interesting, wasn't solving our regression problem. So we went back to the drawing board, and then we tried an approach where we actually found the rotation for which the regression model holds, directly mirroring some of this earlier work in things like partial least squares, though highly tuned to settings where you have a positive definite symmetric matrix outcome. So in principle, what we're saying is we take the logarithm of some rotations and assume a regression model holds, again, assuming common partial commonality. And then we find the rotation and the regressors all in one likelihood-based step. And then again, we also established all the theory for it. I'm going to skip this slide, but we've got fancy equations if you ever want to see them. So just to give you some idea that it works, here's some simulation data where we picked the number of common components when there were actually 2 common components. And you see the measure does a nice jump from 2 to 3, which is what you would hope. And then we hoped that we would get some kind of consistency of the model coefficients as the number of subjects gets larger and larger. And we do see that, which is a good thing, simply because in these kinds of models, we're estimating a lot of things that could perturb the regression relationship, and it's not obvious that would all work. It does stand to reason, I should say, that you do need a lot of subjects for this to work, and then I'm about to apply it to a data set with 13 subjects. The experiment we tried it on, and you'll see the reason why we tried it on this experiment was an aphasia experiment, where they used transcranial direct stimulation versus sham on subjects with primary progressive aphasia. So everyone had aphasia, 15 were logopenic, 13 non-fluent, 8 semantic, but only half were randomized to the treatment, and the other half were randomized to the control. The reason why this is a good setting for something like this is because there's a lot of patients. There's not a lot of patients, but there's a lot of regions. You do want something that reduces your dimensionality, because you want something that tries to eliminate the multiplicity problem. But more than anything, we have a setting here where we know there should be some perturbation in connectivity because of the treatment. The transcranial direct stimulation should have actually perturbed the connectivity. So we know that we should see an effect here up to variation, of course. Then the sample size might be too low. So here's the kinds of things that the method, I'm sorry, here, let me just show we also restricted regions. And in this case, we restricted our region to the area of stimulation, which was the IFG, and then areas that were sort of putatively associated with language and associated with what our collaborators thought should be associated with language recovery and aphasia. So this is what the method gives you, is it gives you a series of weightings on the regions, and then associated with each component. So this is one component. It gives you the regression model. So in this case, the regression model is plotted by the regressor on the horizontal axis and the regression coefficient on the vertical axis. And then because we're doing some sort of statistical inference, we get confidence intervals around them. And here for this component, the only one that was significant was the TDCS versus sham comparison. So you get one such picture like this for every single for every single coefficient, for every single common principal component that you estimate. Of course, you have to be careful with your inference in that you are, in a sense, overfitting the regression model. If you have data unrelated to the regression model, it's still going to try to find the components that that fit the regression model the best. So I think the interpretation of these coefficients is quite confusing, and so I have this kind of simple explanation because we are very concerned with interpretation. So if you look at here's the regions on the vertical axis here, and the colors are the weightings of the principal component vector. So the idea here is that ROIs with the same sign, either both red or both blue because of the sign invariance of principal component vectors, then you would interpret it, you would give it a regression interpretation, like all else being held constant, including the other principal components, increasing the regressor by 1 unit suggests an increase in connectivity between those regions. And then ROIs with different signs, one red, one blue, regardless of which one is red and which one is being blue. All else being held constant, including other components, increasing X suggests a decrease in connectivity between these regions. And of course, the interpretation reverses itself if the coefficient is negative. So it wouldn't be an increase in the top statement, it would be a decrease, and it wouldn't be a decrease, it would be an increase in the bottom statement if the coefficient itself was positive. So you do have to be careful in interpreting these things about all of the sort of double negatives flying around, but I think this is the simple rule we came up with. We do have an R package if you want to use it. It's on Neuroconductor, which is our continuous integration service and package repository for R related resources associated with neuroimaging, and we call it CAP, or covariate assisted principal component regression. You can extend this to whole brain data if you're willing to do an ICA decomposition first. So we've done that. This is just a fancy picture to say that statement. And then we have also extended it to discrete regions, but for a much higher dimensional number of them. The issue, of course, is if you have even just a couple hundred regions, you wind up with a couple thousand region pairs. And so you wind up with a fitting problem. So we have a penalized version of this for higher numbers of regions. And then here's I just wanted to show you that we also tried this on instances where we have a sufficient data actually perform the estimation. So this is an example of applying it on the ADNI data. In my last couple of minutes, and then I'm just focusing right here on the specific sub areas of interest. Given that I'm almost running out of time, I have one last thing that I would like to talk about, which is a entirely different way to think about connectivity regression, where you have connectivity as an outcome. So I really loved Rick's talk, and I think it set this up a little bit in the sense that imagine that you weren't so concerned with localization of the effect at the nodal level. Imagine you were more concerned with just some overall effects. Then, of course, you could consider overall effects within regions or within groups of nodes or something like that. So imagine you take your fMRI, you take your parcellation, you take the bold signals within those parcellations, the average time course within those parcellations, you make your connectivity matrix, and then you simply take the connectivity matrix, and instead of viewing it separately, edge at a time or node pair at a time, you simply take the entire collection of these edges and evaluate the density associated with them. And again, this might make sense in the TDCS application just because you're stimulating a region, and that would have potentially a large regional effect. So our idea was thinking about how can we then use densities, what we call the connectivity density, which is just the large collection of edges from connectivity treated as a function. How can we use that as an outcome itself? So now, instead of having a positive definite symmetric matrix as an outcome, we have a function as an outcome. which is well studied in statistics. There's lots of ways to do it, but one of the most convenient ways to study functions as outcomes, or I'm sorry, one of the most convenient ways to study functions is to treat them as regressors. So we flipped the regressor response relationship. So this is a very common statistical technique in so-called retrospective case control studies. So the famous example is the work of Cornfield 80 years ago or so, where the concern was where you took patients with, say, lung cancer and then matched controls. And because it takes so long to follow an exposure like smoking over time, you, instead of doing a prospective study where you observe people's smoking behavior and then follow prospectively outcomes, you look retrospectively at patients with lung cancer and matched controls, and look retrospectively at their prior smoking behavior. That kind of retrospective case control study flips the regressor response relationship, where you actually know you by virtue of the study design fixed that, let's say you have 100 smokers and 100 matched controls. You then analyze those outcomes as if they were random, or as if they were random. And so we did this exactly with the TDCS data, where we analyzed their patient's outcome status, which is 1 if they were randomized to TDCS, and 0 if they were a control. We analyzed that as the outcome, even though it was fixed to be half and half by the study design. And the mathematical argument as to why you can do that is a simple Bayes rule argument, namely that the odds of the outcome is equal to the prior odds given our model times the prior prior odds of treatment assignment. But in this case, because there was randomization, the prior odds of treatment assignment drops out. And so you get this relationship right here. And so instead of modeling the connectivity as an outcome, we model the treatment status as an outcome. And it all works if you're willing to do a logit model and presume that the logit model holds. So in this case, we have the logit of the probability of receiving a treatment depends on whatever other regressors you're interested in putting in, plus your connectivity density. And our model that we then thought of to put in was, linear with respect to the confounding variables, and then an integral with respect to some function of the connectivity density. And then the goal is to find this coefficient function G. Then the interesting part of this was looking at, both theoretically and practically, what were the different versions of T that you could look at. And we arrived at the natural logarithm of the connectivity density evaluated at the inverse of the quantile function, which is the, let me see if I can actually say this without messing up once, the negative of the log of the derivative of the quantile function inverted. The reason for this is that it tends to focus interest in the tails of the quantile density, where you would expect the tails of the connectivity density, where you would expect the effect to happen. So it's asking this coefficient function not to work so hard, and then taking the logarithm just seems to help regularize it a bit. So our version of principal components regression simply used the connectivity density as the regressor. It then used ordinary functional regression techniques to do that. And so just because I have 30 seconds left, I wanted to show you the kind of things that you get out of this. What you get out of it is a coefficient function that tells you how your connectivity density relates to the log odds, in this case, of receiving treatment. So here we looked at different strategies, and you can see the impact of the choice of that the connectivity density, the choice of that transformation, how big of an effect it has. If you just kind of put the connectivity densities indirectly, you don't see any effect, but this is the TDCS data where we know there should be effect. However, if you put in the log quantile transform or the log quantile transform where the densities are estimated in a different way, you see the effect, but not only do you see the effect, you see the effect in the upper quantiles where you would expect to see the effect, where you would expect to see the connectivity density change between the two groups. So some future directions in this work, we're thinking about dynamic connectivity with dynamic eigenvalues. A student in applied math is thinking about that named Guadong. Yi has already worked on whole brain covariance regression. We have some testing ideas we'd like to look at, thinking about graphs as outcome rather than connectivity densities, and path analysis models. And then I just wanted to share some references here. You can find this talk at my website.